Good afternoon, everybody. It is a pleasure to have all of you here today attending the 8th FNIP event. I'm Letizia Mariotti. I'm a scientist from the Institute of Neuroscience of the CNR. And today I'm going to be here with Dr. Marco Brondi from the Institute of Neuroscience as well, and Federico Rossi from the Italian Institute of Technology in Rovereto. Thanks again for joining us today. So before starting, let's see if okay, yes. Before starting to introduce the amazing speaker we have here today, I would like to introduce you what the FNIP initiative is. So FNIP stands for Fluorescence Neuroimaging and Photonics Program, and it has born with the aim to bring together scientists and researchers who share the interest in the field of advanced microscopy, imaging, and photonics to ultimately study the brain. And we would like to explore these elements at 3 different levels: the photonics level, the T1, the microscopy level, the T2, and the data analysis level, the T3. Behind the organization of the webinars, we are currently 7 young researchers from 4 different departments and institutions here in Padova. In particular, we have Gianluca from the Department of Physics and Astronomy here, and also Nicoletta from the Biology Department, Marco, Jacopo, and Claudia from the Department of Biomedical Science, and finally, myself and Marco from the Institute of Neuroscience of the CNR. So far, almost 30 speakers from all over the world joined this initiative during the past webinars and events, contributing with a lecture and sharing with us their expertise, and we are very grateful to them for their contribution. If you are interested also in the FNIP program, please connect to the official website, where you can find information on the past events here. I don't know if you can see the pointer. You will find all the speakers and the recorded talks, and you can also be updated with the upcoming initiative. In addition, you can contact any one of us in case you are interested in joining this initiative as a speaker, as an organizer, or a supporter, and you can find our contact on the website. Before starting this session, a few important details to participate, to actively participate to this webinar. So in case you want to ask any questions, please use the question and answer button, and you can write down there your question, and we will try to address as many questions as possible at the end of each talk. And if we will not have enough time to read the questions, don't worry. The speaker will have the possibility to reply to your questions by writing down the answer. And at the end of the 3 presentations, there will be a follow-up session where everyone is invited to participate. Oral questions there are allowed, and please raise your hand, clicking that button, and we will enable your microphone. We will try to give priority to PhD students and young researchers, so don't be shy and take this opportunity to interact with the expert that we invited today. So now, if you don't have any questions for this first one, we can, I don't think so. So we can start by introducing the speakers of today. So we are very lucky because we have an exceptional lineup of speakers who will tell us about the latest developments in optical tools to manipulate brain activity. The first speaker of today will be Dr. Perea from the Cajal Institute in Spain. Then, unfortunately, Dr. Robinson could not participate today for personal reasons, so I really want to thank Edgar Bolmer from UCL in the UK, who accepted to be with us today, even with a very short notice. And finally, Dr. Pisoni from the Pasteur Institute in France. So, and now I'll introduce Dr. Perea. So, I'm delighted to have you here today. Thanks for being here with us. Dr. Perea is the head of Neuroglial Network Lab at the Cajal Institute, where her group studied the biological properties that govern neuron-astrocytes interaction in the central nervous system. They use cutting-edge techniques such as ex vivo and in vivo electrophysiology, but also calcium imaging, optogenetic, and chemogenetic approaches to unmask the specific roles of astrocytes in synaptic transmission and behavior. Their goal is to understand the contribution of astrocytes to the information processing by neural circuits and the impact that neuron-astrocyte signaling has on different brain function, either in physiological condition as well as in different brain pathology. And today, her talk will focus on melanopsin for time-controlling activation of astrocyte neuron networks. And thanks again for being here. And now the stage is yours. So if you want to share your presentation, you're free to do it.
Well, thank you so much to the organizers to have the opportunity to participate in this webinar. I'm very happy and thankful for the invitation. So, well, today I'm gonna show you some summary of the data we have in the lab regarding the use of optical tools to manipulate the astrocyte calcium signaling in order to evaluate the consequences for synaptic transmission and behavior of the animal. Well, I'm going to focus on melanopsin. This is a new opsin in the toolbox of opsins available for glial cells as a time-controlling tool to manipulate these astrocyte neural networks. Well, many of you maybe know that the cells we have like 190,000 million cells in the human brain, but maybe not all of you knows that almost half of this amount correspond to glial cells. They are not neurons, they are other type of glial cells in the brain. So when we talk about a nervous system or networks, we are usually thinking neurons and neuronal networks. And this is a picture, confocal pictures taken from hippocampus. And you can see this is the pyramidal cells in the CA1 area of the hippocampus. But it's important to note that behind these neuronal networks, there are also glial networks. And this is the same image, the same field of view, but now you are seeing astrocytes. These green cells are labeled with the GCAM protein. It's a fluorescence protein that can sense also calcium. But you can see that on top of these neuronal networks, there are also other cell types, and in this particular are astrocytes. Astrocytes are one of the most abundant glial cells in the central nervous system, and they have the particularity that they do not overlap their territories. As you can see here, this is an image in which the red astrocyte has been filled with a red fluorescence, and the other cells, the other astrocytes, have been filled independently with the green ones. And you can see there is a very few regions for the overlapping of these processes. So they can take care of their own territories without sharing so much with other astrocytes. The important point of these particular cells, they are facing synapses. They are facing and they are monitoring synaptic transmission. They are taking care of the synapse in the maturation, in the stabilization, and also in the development of the synapse. This is a picture, electron microscopy picture, and you can see in green are labeled the presynaptic component, and you can see the vesicles ready to be released, facing the postsynaptic terminal, so the postsynaptic membrane, and in blue they are fill out the astrocytic processes, and you can see they are just touching very close to the synapse, so and they're really placed to monitor the synaptic transmission. So you can see here this the tripartite synapse that we are called and is well established now in neurophysiology. One of the main feature of these glial cells are not electrical excitable like a neuron. As you can see here, you can see I can use the pointer. Okay. I want to use a pointer, but okay. So you can see here a typical action potential discharge when the neuron is depolarized, and you can see the typical firing of the neurons, the pyramidal cell, and the other is in black, and in red is the same protocol, but now is applied to an astrocyte that are being recorded simultaneously. And you can see that even increasing the steps of depolarization pulse, you cannot evoke or you cannot see any active current in these cells. So they are silent for the electrophysiological point of view. So one of the main reasons that astrocytes have been, let's say, being forget for many years in the study of the nervous system. However, sorry, it's not working. Yeah. However, these cells are not electrically excitable, but they express excitability as other cell types based on calcium signaling. And you can see here, this is a slice, it's a hippocampal slice loaded with a calcium sensor. And in response to a particular stimuli, astrocyte can increase the calcium. You can see an increase in the fluorescence, meaning there is an increase in intracellular calcium of these cells, though they are is a readout of the excitability of these astrocytes. So the main route or the main pathway responsible for this calcium signaling is due to the activation of intracellular stores. So through the metabotropic receptors, maybe different type of neurotransmitters that are linked to GQ or GI, GQ pathway that activate PLC, IP3, and calcium increase from the intracellular stores like reticulum. So this is the main or the canonical way that astrocytes increase the calcium, but there are other routes that contribute even simultaneously to this calcium signaling. These calcium dynamics are very complex, has been reported in the last years with the advantage of two-photon microscopy. And you can see here, this is a process of the astrocytes that is spontaneously having calcium increases. So they are sensing or releasing calcium in particular spots, and can be independent, or you can see that sometimes there are like a huge influx of calcium that can travel to the main process. This activity has been studied for a long time, and now with the super resolution microscopy and 3D imaging, it's possible to reconstruct the activity in an entire process in real time to understand how complex is this signaling. Well, has been proposed or hypothesized that depending on the nature of this calcium signaling and the location and how it propagates into the astrocyte can potentially have different impact for synaptic transmission. Can modulate a very local synapse, can modulate different but close synapses, or can be a global modulation that can even modulate thousands of synapses simultaneously. It's well established that calcium is the main source of the excitability for the astrocytes that can be induced or can be evoked by different actors, in particular neurotransmitters. And when the calcium is increased in these cells, is also able to release or to link the release of different active substances, as you can see here, like ITP or adenosine, even glutamate, GABA, glycerin, etc. Nitric oxide, different molecules that can have an impact in synaptic transmission and neuronal excitability. Well, just a very brief example of this impact is that this calcium signaling is able to modulate at the level of single synapse. So this the tripartite synapse can modulate the synaptic activity, increasing the probability of release of this particular synapse. You can see here we are recording hippocampal neurons, pyramidal neurons in CA1, and also simultaneously recording one single astrocyte. And we evoke or impose calcium signaling in this particular astrocyte and monitor what happened with the synaptic transmission that are receiving this particular cell type, this particular neuron. And we saw that in particular, under particular experimental condition that allows to reduce the intensity of stimulation to isolate one single synapse or very few single synapse, you can see there is an increase in the excitability of this synapse, increasing the efficacy of the synaptic transmission due to an increase in the probability of release. It's a very particular effect that takes place in the presynaptic locus of this synapse. Well, this with this information during the last 20 years, is well established in the field that the synapses are not working, are not operating alone. They need, and they are composed by the other elements, in particular astrocyte, to establish this tripartite synapse. Well, astrocyte can respond to the neuronal activity, can increase the calcium, and then turn back a message to this particular synapse or other synapses to manipulate or to control the synaptic transmission. But these tripartite synapses are very complex, are very flexible, and very plastic, in the sense that can be very heterogeneous, depending on the brain area we're looking at, depending of even in a brain area, a particular regions of this brain area, let's say hippocampus or brain cortex, different layers of the cortex. They can respond, or they can have different activities depending of the rhythm of the network, let's say oscillation. Depending on the cell type we are looking at, excitatory neurons, GABAergic neurons, dopaminergic neurons, etc., and also neurotransmitters that are releasing, and the glial transmitter that astrocyte can release. So this heterogeneous is quite complex, and we need to find ways and tools to decipher what is the particular aspect we are looking at. And I wanted also to mention very briefly that this complexity is even larger if we are looking at the phylogenetic scale, or phylogenetic point of view. Main of the information we have about these tripartite synapses is coming from rodents, but now we have some hints also from human tissue, from human brain, and we know that even there is a huge difference between astrocytes from mouse and human brains. Not only at the level of complexity or number of neurons, but also the complexity and number of the glial cells, and in particular astrocytes. Here I show you one image. This is a mouse image labeled with this marker specific for astrocyte GFAP. And on the side, you can see the same GFAP positive cells in the human brain as a huge. It's not only the size of the soma, but also the complexity, the number and complexity of the ramifications of the processes. As you can see here, that also the amount of the amount of synapses that one single astrocyte can control or can manipulate has been estimated that in mouse brain, one astrocyte can cover up to 1,000 synapses. But in the human brain, one single astrocyte can cover up to 2 million synapses. So the power of one single astrocyte to modulate and to regulate the tune of synaptic transmission in 2 million synapses is quite powerful. So there are different ways to evaluate these neuron-astrocyte networks. There is a bottom-up approach of top-down approach to looking at the synapses, local networks of brain circuits. But in this every step, you need to find the right tools, the right approaches to ask the question you want to answer. And optogenetics, because this webinar is about optical tool, we are looking at, or we are taking advantage of optogenetics that was primarily applied for neurons, but also it's possible to apply this tool for manipulate the astrocyte calcium signaling. And this particular tool, I'm going to mention Channelrhodopsin to put in the context of Melanopsin to evaluate what is the difference between the both opsins. Channelrhodopsin is an ion channel sensitive to light. As you can see here, is permeable to different ions, sodium, potassium, calcium, and also protons. And we apply this tool to manipulate the activity of astrocytes that has been very successful by different labs to control the activity of these astrocytes. So we can use viral approaches, we can use transgenic mice to express in a specific way, in a specific manner, this opsin only in astrocyte and to evaluate what is the consequences for the circuit. We know that stimulating Channelrhodopsin, you can increase calcium signaling, as you can see here in the traces. And this calcium signaling inducing by Channelrhodopsin is able to induce glial transmission, induce to the release of different active substances that modulate synaptic transmission. You can see here in the top that can increase the activity of spontaneous synaptic transmission, excitatory one, but also the activity of inhibitory synaptic transmission. So this tool or this opsin is able to, at the level of the synapse, to modulate the synaptic transmission. So, but it's not the ability of this tool or the power of this tool is that you can also manipulate in vivo activity. So we apply this approach in visual primary cortex, and you can see here where when the animal is anesthetized and is subjected to see or to respond to different visual stimuli, and we are encoding this, or we are detecting this orientation tuning curves in particular cell types of V1. When we apply the approach of stimulating with Channelrhodopsin astrocyte in this particular network, we observe that this, in green, for the excitatory cell, there is an increase and enhancement of the visual properties. But these responses are different if you are looking at excitatory cells or interneurons, and even different type of interneurons, giving the idea of this complexity and heterogeneity of the relationship between astrocyte and neurons. So the summary is this tool allows you to underscore what is the role of astrocytes for a particular brain function. However, Channelrhodopsin, as mentioned, is an ion channel and allows the influx of different ions, so cations in particular, but doesn't mimics or doesn't represent the main route of calcium signaling in astrocyte, that is metabotropic, that is a G protein coupled signaling that link the calcium increase from intercellular stores. So that's the idea that that we were looking for a G protein, but light sensitive protein that can be activated and used in astrocytes. And we face Melanopsin. That Melanopsin is a photopigment that is expressed in the mammalian brain, in the mammalian retina. There is a particular subset of ganglion cells that express Melanopsin. And this expression of this photopigment has been related with the different activities, not related with visual guide function, but also very important for circadian rhythms, for example, for saccadic movements or pupillary light reflex. So there is a endogenous protein in the mammalian brain that is maybe can be useful to take this particular tool and manipulate and genetically manipulate to be expressed only in astrocyte. Well, we took this approach and we take this protein, we fuse with this promoter, the GFAP, that is more predominant expressed in astrocytes, so it's predominant for astrocytes. So we are using this approach we fuse in a plasmid encapsulated in a virus, and we inject mouse with this virus. And we tested if this melanopsin expressed in astrocyte can, increase the calcium when we stimulate with blue light. And we found that this particular opsin can be successfully expressed in astrocytes, specifically in astrocyte, but also we observed that the stimulation of melanopsin with blue light, that is sensitive to blue light, is able to increase the calcium signal at the level of the soma, but also microdomains that we mentioned that are very active and very dynamic. So in both compartments, melanopsin can increase calcium signaling. So we first did like a dose response, so different duration pulses, and evaluate how is the range, the dynamic range to evoke the calcium signals. This is, from 1 to 20 seconds, 1, 5, 10, and 20 seconds of blue light stimulation, and you see there is a run-up responses at the macrodomains levels and also at the soma. But what were the consequences? We can use this tool to evaluate the properties of the associated neural networks. Well, the answer is yes. This tool allows to increase calcium signaling, and we saw before, but also we can evaluate the consequences for the network if we are recording neurons and stimulate the astrocytes that were expressing melanopsin. You can see there is an increase, a transient increase of synaptic transmission, excitatory synaptic transmission. So this tool able to increase the calcium, but also stimulate glial transmission. Well, we wanted to evaluate if this tool is somehow mimicking or recapitulating the endogenous calcium signaling in astrocytes. And we know that melanopsin is linked to GQ and also GI, but mainly GQ proteins that give rise to calcium signaling through the IP3 production. So we take advantage of these knockout mice, IP3 knockout mice that has a downregulated calcium signaling in astrocyte, but we express melanopsin in these cells and apply the blue light now. So if the melanopsin is linked and positive link to the GQ and increasing the production of IP3 and calcium release from intracellular stores, we should observe a calcium increase in these animals. However, these animals are knockout for this particular receptor 2 of the IP3. So they cannot release calcium through this mechanism. Then when we stimulate with the light, melanopsin was not able to produce or to stimulate the calcium signal through the release of calcium from the intracellular stores, suggesting that they share the common mechanism. So melanopsin is indeed acting through the canonical pathway that is also expressed in the mammalian ganglion cells. So we can recapitulate the intracellular calcium signal, so suggesting that this is a very like an optimal tool to study the metabotropic activation of astrocytes through synaptic transmission, for example. Another point here that we wanted to evaluate is, okay, we are using different duration of the light from 1 to 20 seconds to see the dynamic range of this signal. But the brain, usually is working at different range of brain oscillation, different dynamic ranges. So you can see this is an EEG, human EEG that shows a very wide spectrum of different frequencies, from very super slow oscillation to these ultra fast oscillations. So 50 to 40 seconds to see one event, or 2000, 1000 hertz to find an event. So there are wide spectrum of the activities that you can find in the EEG human brain. However, we are going to focus on these particular ultra slow brain oscillations because they are quite similar to the intrinsic oscillatory activity of the astrocytes at the soma, looking at the soma of the astrocyte, when we found at the ex vivo samples, no, brain slices. So we observed that the soma can oscillate in this range, 0.05 hertz, that is in the range of the 5 to 15 seconds, 15 to 45 seconds EEG signal. We are using this endogenous frequency, and we apply melanopsin with the simulation of light to recapitulate this endogenous signaling. So 0.05 hertz of light. We are applying these pulses, as you can see here, we call low frequency simulation, and we evaluate what is the consequences for astrocytes and synaptic network. And you can see here, in that condition, there is no transient effect. We observe long-lasting effect. We observe LTP, long-term potentiation of synaptic transmission. That is one of the cellular mechanisms hypothesized underlying memory and learning. So applying this low-frequency stimulation to astrocytes that express melanopsin is possible to transform a signal that was transient in a long-lasting response. So we found a way that melanopsin allows you to change the activity range of the astrocyte to play with these frequencies from sustained activation and low frequency stimulation, and you can get from transient to long-lasting synaptic changes, even or at least working with the same molecule. We found a way to manipulate the same cells, just changing the activity pattern. We can modulate also what is the output for the network. And what is the consequences for the behavior of the animals? We apply this tool, or we study this tool in ex vivo preparation, but the final goal is to evaluate what is the consequences of this astrocyte activity for brain behavior. And we apply this tool in different behavioral paradigms, and we are showing here just two, the goal-directed behaviors, and also the novel object location that are linked to, one is prefrontal cortex mainly, or main rule for by prefrontal cortex, and the second, the novel object location, is mainly ruled by hippocampus. So expressing melanopsin in these particular regions is possible, just activating astrocytes to modulate the synaptic, not only the synaptic function, but also the brain behavior, the animal behavior. And you can see here, in using melanopsin, we can increase or improve the cognitive performance of these animals. They can perform much better in the T-maze, so they can make correct choices when we're stimulated with melanopsin, and they can remember better when they were stimulated with melanopsin. So in both conditions are associated the triggers of this improvement. But because we were starting the talk about talking about Channelrhodopsin and now Melanopsin, we wanted to evaluate the difference. Okay, very basic approach, just to evaluate what are the main difference or the main common points of these particular tools. Because both opsins are increasing calcium, are stimulating with blue light, and are, at least Channelrhodopsin is widely used for astrocytes. So we are comparing here 1 second to 20-second light stimulation for Melanopsin and Channelrhodopsin, and you can see here in blue, or like a light blue is Melanopsin, orange Channelrhodopsin. You can see for 1 second, Melanopsin was able to increase calcium signals, to increase the frequency, to detect more events in the calcium signals than Channelrhodopsin. So it's more sensitive, the Melanopsin, as has been reported for the in retinal cells, is more sensitive to the blue light, and that maybe that's the reason that with short pulses is able to increase calcium, while Channelrhodopsin needs at least 5-10 seconds to increase calcium signals in astrocytes, or at least to detect in our hands. And also in parallel, we studied the consequences for synaptic transmission. And you can see here from 1 second to 20 seconds, 1 second is not able to, or is not enough to see like a global effect for synaptic transmission. Even the calcium was increased, but we are looking at a huge amount of synapse. It's not single synapse stimulation. So can be dilute, can be underestimated if we are looking at a bulk condition. However, if we are looking at the 20-second, and both opsins are able to increase calcium and evoke synaptic changes, you can see that Melanopsin induces like a transient effect, while Channelrhodopsin induces like a long-lasting effect. So with this tool, even using the same duration of the light pulse, we lose the dynamic or the temporal control of the synaptic responses. So depending on what is your question, depending what is the goal that you are want to reach, you need to find the right tool. Maybe Channelrhodopsin or Melanopsin is much better depending on the region, the type of circuit you are looking at, and the type of responses you are studying. And it's also important to note that we found that the same different opsins can release different gliotransmitters. Even when we are looking at the consequences for astrocyte or for a cell, if you are looking or thinking about neuron, you are thinking about action potential, so fighting discharge. And in the case of astrocytes, we are looking at calcium signaling. The calcium signaling is a readout, so to validate the tool. Both opsins can increase calcium. However, this calcium for sure means something different for the cells, because in one case, they are releasing glutamate, and with Channelrhodopsin and Melanopsin release purines, release ATP or adenosine. So it's important to note that the calcium was increased in both cases, but for intracellular point of view, this calcium has a different meaning. In one case, it's releasing glutamate. In the other case, it's releasing ATP, adenosine. So our home message that I want to just make an effort to clarify this point is Melanopsin can be a useful tool to study neuron-astrocyte networks, and we provide evidence that this tool can mimic the endogenous signaling pathway triggered by G protein that is linked to the synaptic transmission or the synaptic activity. So can be like a better tool depending on what is your question again, to evaluate these neuron-astrocyte networks. And finally, I want to thank my team, the Neuron Glial Network Labs at the Cajal Institute, and in particular, Sara Mederos, that was a former PhD, that was making most of the results that I presented today. So thank you so much, and I'm open for your questions.